My name is Allison Brent. I am honored to serve as your Chief Science Officer for the Foundation for Angelman Syndrome Therapeutics. Okay, so just a brief intro of my roles here at FAST and in the Angelman community. So I am the CSO for FAST. I also serve as the Chief Operating Officer of Genetics Biotherapeutics. I am the Director of the Angelman Syndrome Biomarker and Outcome Measure Consortium. I am the co-director of InSync, the International Angelman Syndrome Research Council, which is a new initiative that we started this year that has been really exciting and has really helped to really bring in some new blood, new ideas, new people into the Angelman space in order to really help us understand our gaps, and I'll talk a little bit more about that. I am also a veterinarian. Like I had said earlier, I'm an internist and an interventionalist, and I mostly do minimally invasive surgery, developing medical devices for animals with naturally occurring diseases. And that's the only career I ever wanted, actually since I was 8, until 2015, when this beautiful little girl was diagnosed with Angelman syndrome. So what I'm going to do today is really talk about Angelman syndrome, and for all the speakers in the room for the rest of the day, I'm going to do the best I can, which probably isn't as good as what you can do, to explain what Angelman syndrome is to our community and to our families and to everyone watching both in the room and online. So what I want to ask all of you speakers to follow is that you don't need to do this again, because they're really, really, really smart and very sophisticated, and they get it. They get what Angelman syndrome is, they get the genetics behind it. So if your slides have a lot of Angelman syndrome, what is it and what are the symptoms, I promise you, we know the symptoms of Angelman syndrome. You don't need to remind us. That is for sure. But if there's anything I leave out that you think you could have done, that I should have added, please do include that in your slides and in your presentation. But we don't need to go over Angelman syndrome again, because this team really gets it. But what I want to explain to everyone in the room is a little bit about the genetics behind Angelman. It's really important that you understand this. We are in a place right now where disease-modifying therapies, gene therapy, gene editing, is happening for our children. And if you don't understand this and you're not able to help make the best decisions for your child or your loved one, then you're not going to be in a place to feel like you have the power to control what happens to the future of your child. Now, none of us have the power to control it. We're all, jumping in head first. But ultimately, the power is knowledge, and the more you understand and the more you really grasp and the more questions you ask, so don't be shy, the better you're going to have a grasp of what the options are for you and your child. If you want to take those options, if you are ready to take a risk, because everything we're going to talk about will have a risk. It hopefully will have an amazing benefit. But there's always risk first, which is why clinical trials start with a phase 1, which is safety. And we have to remember that. Everything we're about to talk about for the next 2 days will start with a phase 1, which means that we have to be willing to take that risk. And some of us will be, but some of us won't. And it is absolutely okay if you are not ready to do that and you want to see where it lands and see how it plays out for other people. So nobody in this room, even though we're all about curing Angelman syndrome, need to feel that they need to be patient 1 or need to be patient 5 or even patient 50. So please remember that. But the more you understand and the more you know, the more comfortable you're going to feel making the right decision for your child or your family member. So let's talk a little bit about why Angelman syndrome happens, okay? So when we think about Angelman syndrome, we know that it is a monogenetic disorder. What does that mean? It's a single gene disorder. For kids with deletions, it's not a single gene disorder. It's a 10 gene disorder, maybe 11, maybe 9, maybe 7. But there's one major gene that is common amongst all of our children, and that is they are missing a gene called UBE3A. And they're either missing it or it doesn't work well. So let's talk about that. So in humans, there are 46 chromosomes, or 23 pairs. We get 1 from mom, and we get 1 from dad. And this is what they look like. Here are the 46 chromosomes, or 23 pairs. And in Angelman syndrome, on the 15th chromosome, on the mother's copy, there is a gene that makes a protein that doesn't work, or it's not there. And this impacts approximately 1 in 15,000 kids in the world that live with Angelman syndrome, or so we think. That's what the studies show. The studies are soft, so we don't really know exactly how many children. But if this were true, which so far it seems it is when populations are assessed, and thank you to Terry Jo Bichelle, who worked really hard for an ICD-10 code, we're probably going to know the answer to this sooner than we would have if it weren't for her. So a round of applause to you, Terry Jo, for that. Because we have an ICD-10 code, we're going to be able to identify how many individuals are diagnosed earlier, and we'll be able to really figure that out over a period of time, the incidence in this country, which seems to follow the incidence in every country in which these tests are done. But 1 in 15,000 kids, what does that mean? That means 25,000 people with Angelman syndrome in the United States. That could mean 500,000 kids in the world with Angelman syndrome. Where are they? We need to find them. It is our job to find them, right? So we know that we have probably close to 10 to 12,000 people probably known with Angelman syndrome. We need to find all of them. So we're missing about 490,000 kids. Where are they? It is random, so most of the time this will never happen again, or there's a 1 in 15,000 chance it would happen again to you or to your family. But sometimes it's rarely inherited, in which case you have a 50% chance of having it, and that's typically associated with a mutation. Very, very uncommon mutations can be inherited. And each chromosome, like I said, is made up of many, many genes, thousands and thousands of genes in the genome. And each gene typically makes a protein. And those proteins are what have function for the body. And when you're missing the gene, you are therefore missing a protein. And if you're missing that protein, some proteins, for me, I hate the taste of cilantro. It tastes like soap. That's a genetic disorder. I wish that's what UBE3A did. But unfortunately, as we all know, it is not the cilantro gene that our kids are missing. Okay, so when we think about Angelman syndrome, we have that maternal copy and that paternal copy. Okay, and then let's just make this a little bit simpler in a schematic. So when we have those two copies, what's really, really interesting about this gene, so if there's a shining light or a silver lining, it's the fact that our kids have a single gene disorder that is non-degenerative. That's amazing. And we should be very, very grateful for that, because there are many people, and many of which are in this room with us, so we're very lucky to have all of you with us today. We have a rare disease consortium that's here with us today that doesn't have Angelman, but is learning from what we've done here at FAST in order to help accelerate their own programs. So we're proud to have you all in the room as well. But some of them are living with degenerative disorders. And so we need to be really, really grateful that we're not. Doesn't mean it's not impactful and severely debilitating, but it's not degenerative. And what we also need to be grateful for is the fact that it's incredibly elegant and really important to realize that our kids are missing one copy of the gene on the mother's side. They have a father's copy. And in every single cell of the body, most of our genes are read from mom and from dad, so we have two copies. And if you're missing one copy, that may have absolutely no impact on you clinically. Or it may have a little bit of an impact. Maybe you may walk a little bit differently, or maybe you may have a GI problem because you're what we call haploinsufficient. You have some functioning gene, 50%, the other 50% is not there. In Angelman syndrome, it's not impacted in the rest of their body as far as we know. But why it's impacted in the brain and it's neurologic is because only in the brain it's imprinted. So what that means is the father's copy is silent. Only in neurons of the brain, the cells of the brain. The mother's copy is fully active. So I have one copy working in my brain. You have one copy working in your brain. Because the father's copy is there, but it's silenced by something called the UBE3A antisense transcript. Okay, so that's basically a bunch of RNA, which is a genetic material, that is blocking the father's copy from reading UBE3A. And because of that, they are only reading the mother's copy, all of us. But what happens if there was a problem on the mother's side? Okay, so we have a stop on the father's side, but our kids are missing the mother's side. So what does that mean? That means that in their brain they have 0 UBE3A, but in the rest of their body they have 50%, because the father's copy isn't silent in any other cell but neurons. Does that make sense? So because of that, we are so lucky that we know our kids only need one copy of the gene in their brain, and it's a neurologic-only disorder. Therefore, we have an opportunity to exploit this antisense transcript and activate it, because the gene is there, the gene is healthy, the gene is absolutely able to be activated, because we know the father's copy works in the rest of the body. Therefore, if we can inhibit that antisense transcript and stop the stop, we can turn on the father's copy. And that is the only copy that we would need, because they only need one copy in their brain. Or we can replace the mother's copy. So we have two opportunities here, which is incredibly promising, that most other neurologic disorders do, or genetic disorders do not have. So we're lucky for a couple of reasons. See, I'm always the eternal optimist. That's called an imprinted gene. So how can this present? Well, we can have a deletion of the gene. So meaning that the entire area on the maternal side is missing and it's deleted, or one little letter can be mutated. And that's a very important little letter. But if it is mutated, the protein may not ever be made. So the UBE3A protein may stop in its, what we call its transcription, and it can't translate into a protein, it stops, and it never makes the UBE3A protein. So that's a mutation. It can also result in having 2 copies of the father's side, so 2 antisense transcripts, 2 stops, so 2 very healthy copies of the gene, but they're both behaving like the father's side. So that is UPD, and very similarly ICD, okay? So the most common, 70% or so, have a deletion, but 10% or so have a point mutation, and 3% or so have UPD or ICD. And then there's the really, really rare, the mosaics, who have some cells, maybe 5 to 20, 40%, that have some UBE3A that is normal in those cells, but the rest of the cells don't have it. And then there are some individuals that have never been diagnosed with their cause of Angelman but are clinically diagnosed. And it just probably means the testing hasn't been right for them up until this point, but we may be able to figure out if they have Angelman syndrome now that the testing is so incredibly advanced. Okay, so what does UBE3A do? Well, I promise you, as most of the people in this audience know, it's an incredibly important protein. So what it does is it's an enzyme. So it has a function in that it tags other proteins in the brain in order to tell them how to behave. They're either going to be overexpressed by being tagged, or they're going to be degraded or taken away by being tagged. So it's kind of like the police of the space between 2 neurons. So if we think about that, it essentially allows for what we call homeostasis, or balance in neurons. And neurons usually speak to each other, so there's always a connection of 2 neurons together. And that connection, that space between them where they talk, one neuron has to tell another neuron to do something, and then that neuron tells a muscle to do something. So if I want to walk, my brain says walk, it tells another neuron to walk, and then that neuron tells a muscle to walk. And when it does that, I go to take a step because I'm listening to my first neuron, and then I relax one leg as I activate another leg, right? So I'm going to take a step, relax, and walk. So there's a coordination that happens there of activation, inhibition, and that is a very, very fine skill that's very complicated. We take advantage of that because it comes so naturally to us. But what happens is that if you don't have that coordination in that communication between neurons or neuron and muscle, then you have a really hard time doing things that your brain wants to do. So you want to relax and you can't. You want to activate and you can't. Someone tells you to do something, and the more you want to do it, the harder it is for you to do it. That's called dyspraxia or apraxia. Have I not described your child? Because I pretty much just described mine. So your child can't sleep because their brain's too active. Your child can't relax because their brain's too active. Your child has seizures because their brain is too active. Your child has trouble walking and balancing because that coordination is really, really difficult. Your child can't speak words because your mouth has hundreds and hundreds of muscles that need to be coordinated with your tongue and your voice box and your diaphragm and your breath in order for you to say a single word. Imagine how complicated that is. So actually, the fact that our kids can walk at all, as far as I'm concerned, is a miracle, because it is really hard to walk. And the fact that our kids can is, in my mind, pretty profound. And which is why when they take their first steps, which may be 18 months to 14 years or 15 years old, it is a miracle for all of us because it's really, really hard for them to do. And so this is called a synaptopathy, meaning that the synapse, the junction between those nerves, are really challenged. Okay, so again, I don't need to tell you guys in the room what are the symptoms of Angelman syndrome, but I do think for our researchers and our industry partners that are in the room, I think you should hear a parent tell you what the symptoms of Angelman syndrome are, because you could read it in a book, but I can describe to you what that feels like and what that looks like. And it's important that we all read in books what the symptoms are for our children, right? So obviously they have a universal lack of speech. They have life-threatening and debilitating seizures at times. They have severe developmental delay. They're incoordinated, as I already said. They have ataxia. They have dyspraxia. They can have significant sleep disturbances. Some of my best friends in the room, their kids sleep 2 or 3 hours a night. That is devastating to a family. They can have severe feeding issues. Some require a feeding tube. They can have disruptive or aggressive behaviors at times. And they overall, generally, are absolutely unable to live an independent life. And that is the most devastating part of the disease. But what's the impact that that has on us, on the family, on the people that are in this room and people watching online? Many families can't maintain employment because they have to take care of their loved one with Angelman. The adults, the parents, and the siblings can have significant anxiety, depression, and very, very high stress levels. A lot of that is related to the lack of sleep, and studies have been done showing parent stress is associated with the lack of sleep. Parents have social isolation, impact on family relationships, not only as a couple, but as external family relationships as well, and with their friends. And they really can have a lot of difficulty caring for not only themselves, but the other children and their spouses. And so if you could only imagine the impact of the child, but then the impact of the family. So it is pretty profound, and overall fatigue, which we all can agree with. We are tired. We are very tired. Okay. But what's really exciting? So this paper was published in 2004, and it really stuck with me when Quincy was diagnosed. This is the first paper that I actually read, and it's the first paper that had a huge impact on my view of Angelman syndrome. And this is really important because what this tells us, so this is a paper that looked at a very small population of kids with Angelman that have mosaicism. So what does that mean? I told you earlier, mosaicism is in which individuals with Angelman have some cells that are functional, some cells that have completely normal UB3A. And that can range anywhere from 1% normal cells to 40% normal cells. And so you can see here that those kids that were diagnosed between 1 and 40%, oh, sorry, can you guys go back a slide? Where's the laser? Oh, it's over here. Thank you. So where they have between 1 and 40%, I think the laser is here, you can see that there's a trajectory, and that matches the clinical trajectory. So individuals that have 1% or 0% are down here, and 40% are up here in terms of their clinical score. But what's important to realize is that individuals with 1 to 5% of UBE3A have few to no seizures, are completely ambulatory, and have some speech. My daughter has 0% UBE3A. She's a large deletion. That is not my child. So if you can only imagine what 1 to 5% of UBE3A could do for our kids, we don't need 100% of UBE3A to make a big, big difference in their life. What about 20% of UBE3A? No seizures, fully ambulatory, and can speak in sentences. Are you kidding me? So 20% of UBE3A can give Quincy a life. That is not in the literature. That's pretty impressive. Why is there no child diagnosed with Angelman above 40%? Probably because 41% is considered neurotypical. Okay? So our kids probably don't need more than 40%, and they haven't had a genetic test to be diagnosed because there may be very little wrong, that they never had that genetic testing. So this is what is so promising to me, is that a little bit can go a really, really long way, and that's why I think we have to think about that as we think about therapeutic approaches. Okay, so why are we here? I'm going to speed along now, but I wanted everyone to really understand that, and it's really important you do in order to know where we're going. So let's talk a little bit about our vision and why we're all here. So FAST, as you all know, is an all-volunteer board of parents and professionals. We're obviously all dedicated to one mission, which is transformative treatments for individuals with AS, regardless of their age or genotype, in order to really bring an ultimate cure for our kids. And that is our goal. And I know cure is a tough word, but we're going to talk a lot about cure today. And it's done through a funding strategy that's aggressive, through collaborating, understanding, readying, and expediting drug development in every way that we can. And it's our goal that the programs that we fund really accelerate from proof of concept to human candidate, which will ultimately be leveraged toward a human clinical trial. And we do that through two boards, a scientific board of directors and a parent board of directors. Okay? And so our funding philosophy, really, as I said, we're going to talk a lot about the word cure today, because we're here to cure Angelman syndrome, regardless of what that word means to you. But it has a lot of meanings. And if we break cure down, what it really means is our funding philosophy. And that is that we collaborate. Okay, so we really work toward collaborating with families, with our scientists, with pharmaceutical companies, with our clinicians, with the FAST Global team, with the Angelman Syndrome Foundation, and all of the other Angelman syndrome organizations out there for a single goal, helping other neurodevelopmental disorders along the way in order to share research, organizational updates, and in order to promote and encourage progress toward human clinical trials to support our entire community. We need to understand, okay? So the U in cure is understand. And we want to investigate, identify, and fund research opportunities leading to numerous human therapeutic candidates in order to be able to ensure that we have all shots on goal in order to figure out the best way to treat these patients. And that's obviously incredibly important to FAST. We're here to ready. We want to ready in order to be able to accelerate clinical trial development. So we establish and maintain a framework to ensure that we are testing the most robust drugs in the most robust models, that we design clinical endpoints and biomarkers that are not only sensitive but incredibly meaningful to our kids and our families, and that we're supporting that for all genotypes and all ages, and that we can de-risk all of these programs so that we can get them to clinical trials as quickly as possible in order to understand what works in humans. And then finally, it's to expedite. So we want to accelerate promising research toward clinical trials, approvals, and global patient access, right? That is the most important thing to us, is bringing therapeutics to every single child globally with Angelman syndrome. And we do that through a robust parallel funding strategy. So we really want to be able to work in all areas in order to support drug development for Angelman syndrome. So let me talk a little bit more about all of these parallel strategies as well. And so really just some facts that I think are important for everyone is that Angelman syndrome, like I said earlier, could impact potentially 500,000 kids with Angelman syndrome. I'm sorry, go back again. I apologize. I keep hitting the advance instead of the laser. If you can go back one slide, please. Okay, great, sorry. So 500,000 individuals in the world may have Angelman syndrome. And as John mentioned, FAST was launched in 2008 thanks to many very important people that have been part of the history of FAST, especially Paula Evans, who we are incredibly grateful for her tenacity and everything she did in order to advance FAST and bring FAST really to where it is. FAST has really invested over $25 million to help bring transformative therapeutics to humans living with AS. 23 pharmaceutical companies are known to you, and we're going to talk a lot more because we now counted after yesterday, it's 28 pharmaceutical companies working in Angelman syndrome. Over 20 different academic labs we have funded to support Angelman syndrome research, and in the room listening, either listening in the room today or in the room yesterday, there were 37 different universities being supported by Angelman syndrome research. 9 gene therapy or disease-modifying programs are being funded by FAST in order to accelerate them toward a clinical trial. And we've invested over $3 million to create novel animal models for Angelman syndrome. So when we look at the timelines, it's just really important to realize where we are and where we're going. And this is expediting faster and faster. So as obviously we started in 2008, and a lot of things have happened over this period of time, but ultimately what it culminated in was a huge influx of support that happened about 2013, when the FAST Fire team was funded, which we'll talk a little bit more about, thanks to the Azut family who's in this room, who really believed that collaborative efforts were what was going to accelerate drug development. And we were able to do that. Thank you very much. They're shy and humble, and they don't want to stand, and I've embarrassed him, but I will let him speak for himself. But if it weren't for people like them, no one would believe in bringing all boats higher by collaborating and creating really, really impressive research programs. And that started in 2013. And then really in 2015, we were in a position that we were able to say, we have a whole lot going on and we need to accelerate that further, and we'll talk about that. Where that allowed us to really invest in 2016 in 5 different gene therapy strategies, mainly because we had the hugest influx of funds that we ever had, thanks to the Manier-Lapostale Foundation, where we raised $5.8 million in about 6 weeks of a Cure Angelman Syndrome campaign. And they are also in the room, so I would like to thank them. We all have to remember where we've come from and where we're going. And we're going faster than we've ever gone before, thanks to all of these people. In 2017, because of all the work and because of all of this funding and that huge influx of funds, we were able to launch a biotherapeutic company called Genetics Biotherapeutics. And we did that because we had a promising human candidate that was looking incredible for the potential to go to a clinical trial for Angelman syndrome. And we decided as an organization that we needed to do this ourselves in order to de-risk this further, to be further appealing, so a pharmaceutical company would take this into humans. And what happened? That's exactly what happened. And because we were able to show that this was de-risked and this was promising, we were able to partner this with Ultragenyx Pharmaceuticals to help us accelerate this into human clinical trials. So again, we thank you for that. And we launched that clinical trial, Genetics launched that clinical trial in 2020. And now Roche is in clinical trial, and many others are approaching clinical trial, and we'll talk more about that as well. So we are here to support all. So let's talk about our roadmap for success. So we either need to, like I said, replace the missing copy, the maternal copy of the gene. We need to activate the paternal copy of the gene. We can actually go downstream and we can look at the synapse and we can really address all of the proteins that are abnormal there and think about how we can do some downstream targeting and really make the coordination between those nerves better. And then we must prepare. We have to be prepared for clinical trials. We have to create and characterize these animal models. We have to support biomarkers and outcome measures. We really need to push clinical trial design and ensure that we're supporting all companies to design their clinical trials that are the most appropriate for Angelman syndrome patients, and really be able to ensure we identify patients and realize we're leaving no stone unturned as we think about our therapeutic pipelines. So what are these options? So let's talk about science, okay? So these options, I'm only going to go very briefly through these because all of these people are going to be talking to you today, but I'm going to do it at a high level. So if we do a gene replacement therapy, we can do that through an AAV gene therapy, so UBE3A going into an adeno-associated virus. We could do that through hematopoietic stem cell lentivirus gene therapy, which Dr. Nolta just talked about, and Dr. Obedi and Anderson will be talking about later. And then we can replace the enzyme. So the UBE3A protein, we can potentially put that into the brain. And that alone could have impact on the communication between neurons, giving that protein back where it belongs. And so here are all the companies that are working on a gene or a protein replacement therapy, companies and academic partners. We can activate the paternal copy of the gene, right? We have two options. We're really, really lucky. And so if we activate it or stop the stop, we have antisense oligonucleotides and spherical nucleic acids now in Angelman. We have artificial transcription factors, CRISPR technology, and then we have RNA interference or shRNA and miRNA. We're going to talk about all of these later today and tomorrow. And then downstream targeting, so we can think about the synapse. So let's just talk a little bit about gene replacement therapy. So AAV. AAV is adeno-associated virus. This is a healthy virus. All the bad parts have been taken out, and a very good part has been put in, UBE3A, which is what our kids are missing. And that is delivered into the central nervous system or into the brain in order to what we call transfect or deliver the gene into the neurons so that the gene can be in the neurons, can make the protein, and then those neurons can function with UBE3A in them. And that was first reported out of the University of South Florida by Dr. Ed Weber and Dr. Kevin Nash. And that was in 2011 where they reported this data. UPenn has now also reported data rescuing different things in the mouse model. University of North Carolina, Chapel Hill, in collaboration with Taysha Pharmaceuticals, or Taysha Therapies, they are also working on a UBE3A replacement using 2 isoforms, where they've had a bunch of rescue in the animal model. Joe Anderson just reported the hematopoietic stem cell gene therapy program that was reported in 2021, where they've had a tremendous amount of rescue in the animal model. And there are 4 additional programs being actively pursued by different pharmaceutical companies or academic partners for gene therapy programs. So we're incredibly lucky that we have a lot of shots on goal here. So what does gene therapy look like? So we say you put all this money into these animal models and you put all this money into testing it in animal models, but if you don't get a sense of what that looks like, I feel like it doesn't mean as much to you and you won't understand why we're so excited. So this is just an example. This is a paper that Matt Judson and Ben Philpott just published, basically showing that wild type is a normal mouse without Angelman syndrome, and this is a mouse with Angelman syndrome. And notice how the wild type mouse can, what we call, they can do, basically make a nest. They can create a nest out of paper. So it's called nest building. And without any treatment, Angelman syndrome cannot build a nest. Okay, so that's a complicated task. But with Angelman syndrome, with the AAV or the gene therapy, look how they can build a nest. So I found that pretty impressive, that we can go from not being able to build a nest to building a nest. That's independent life to me, in my mind, for my child. So I look at that and say, that's impressive. That's a difference. Some change has been made. Now look at this. So this is the hematopoietic stem cell gene therapy out of Joe Anderson's lab, and this is the work of Jill Silverman. But look at this mouse model with Angelman syndrome. Okay, so you can see this is a treadmill. This looks like Quincy on the treadmill. I don't know if you've ever seen her on a treadmill, but that's about what it looks like. So they have a hard time coordinating. They have a hard time keeping up. Okay, so they're hitting the bumper nonstop. And then after gene therapy, this is the mouse. Totally different, profound, totally different. And so this is what gene therapy can do for Angelman syndrome. And I think if you don't see this and you don't realize the differences things can make, you don't realize why we're so excited, but also why this costs a lot of money. Because like one of these treadmills for a mouse is like 50 grand. I kid you not. So you have to understand why things are so expensive and why our funding is so important. So let's talk a little bit about enzyme replacement therapy. So this is something we're working on very early stages for Angelman syndrome, but this is for Batten disease, and this is an approved drug called Brineura, which was approved by BioMarin. But ultimately, a port is put right under the scalp, and the enzyme is delivered right into the brain. And this is for Batten disease. But there is some promise that UBE3A could actually benefit outside the cell and also benefit from an enzyme replacement therapy, the protein being directly delivered into the brain. And that is something that we're looking into now, because that could be a therapeutic that is not lifelong. It needs to be repeated, but ultimately could make a huge difference while we wait for all of these gene therapies to come online. So what about activating the father's copy of the gene? So we have many, many different ways that could be done, but really the first person to ever recognize that this was an option was Dr. Ben Philpott at the University of North Carolina, Chapel Hill. He published in 2011 the use of topotecan, which is a topoisomerase inhibitor or a type of chemotherapy, could knock down the antisense transcript and activate the paternal copy of the gene. But that wasn't able to be done clinically in a rodent model because it was so toxic. So we didn't know what the results of that would look like from the behavior rescue. And so then Dr. Art Beaudet was the first to publish looking at antisense oligonucleotides in 2015 in the mouse model of Angelman syndrome. And he was able to rescue some of the phenotypes of Angelman syndrome in the adult mouse model, which we'll talk about. And then Dr. Dindo in 2021 published also looking at an antisense oligonucleotide for Angelman. And then more recently, Roche just published another candidate of ASO in a mouse model, rescuing phenotype in Angelman, which we'll talk about. Dave Siegel published Artificial Transcription Factors. It was Barb Baylis's publication, who's sitting right here, through the guidances of Dave Siegel in 2016. Ryan Butler and Stormy Chamberlain are both working on an shRNA approach for Angelman syndrome, and Jim Wilson and his team are working on an miRNA approach for Angelman syndrome. So we have so many shots on goal to activate that father's copy. We are so lucky. And so these are all the different programs here, but you can see from Art Beaudet's program that they were able to knock down the antisense by about 60%. They were able to rescue some of the cognitive deficits in the adult mouse model. And they accomplished that by injecting this directly into the brain through the ICV, which is the ventricles of the brain. And then basically right now we have 2 clinical trials in humans that are underway, Genetics in collaboration with Ultragenyx, as well as Roche in collaboration with Genentech. In the United States and in Europe. And so this is incredibly exciting that we have 2 and hopefully more coming online for paternal gene activation. And this is a paper that came out of the artificial transcription factor. This is Barb's paper. And what you can see here is the only thing you have to realize is that dark is bad and bright is good. Right? So we are activating the father's copy of the gene when it turns bright red. We are not activating the father's copy of the gene when it's not bright red. So when you see something turn brighter, that usually means that's good. And so really what this is saying is that we can activate that and we can really turn on the father's copy. But this is a little bit less specific than something like an ASO, which is why this is not really pushing as far ahead as some of these other more specific approaches. And this was Mark Zilka's work out of University of North Carolina looking at CRISPR technology. And he was able to show that he was able to rescue phenotype in the mouse model of Angelman syndrome when this was given very early in development. And he was able to rescue many of the symptoms that the mice have of Angelman syndrome. And then we can't forget about the downstream targets because they can be quite impactful as well. And so when we think about that, gaboxidol, which was Ovid's drug, that unfortunately just failed the phase 3 of the clinical trial, but that was really meant to target this area right here. Ketone supplements, disruptive nutrition, they did a clinical trial on ketone supplements, as well as IGF-1 and IGF-2 ligands or agonists. And that was done out of NYU, and now Neuron Pharmaceuticals is working on an IGF-1 analog, which we'll talk about. And so this is Neuron's program, NNZ2591, which is hopefully going to be in clinical trial in the very, very near term in Australia. And really what they were able to see is a tremendous rescue in many of the activities in a mouse with Angelman syndrome. And this is essentially a pill or a liquid that can be given, and that can really, the hope is that it could potentially regulate the synapse between neurons so that they can speak to each other in a better way. So all you need to look at here, you don't have to read it because I know it's a busy slide. All you need to look at here is the red and the green. And what I want you to appreciate here are these are all of the studies that I just talked about. And these are all of the studies and where there was rescue in different tests in the mouse model of Angelman syndrome. But I think what you also need to realize is that our children are not mice. So it is an incredible decade. It has been an incredible decade to be a mouse with Angelman syndrome because we've cured symptoms in many different ways. But what you also need to realize is that a lot of this data is contradictory, right? So there's a lot of red over here as the mice get older, different things are rescued. And it does show that as the mouse gets older, you rescue less things. But some of this work here shows in an adult that you can rescue everything. Some of this work here in adult shows that you can rescue things here that you couldn't rescue here. So some of this work is contradictory, and that's just because the animal model is not perfect. And so we can't get hung up on what is the critical window in age. What we need to get hung up on is that Angelman syndrome can be treated potentially with different ways to activate the gene or downstream targeting. And we can only know so much in the mouse, and then we have to quickly get out of the mouse and start figuring out what we can do in humans. Because as human data comes online, only they are going to tell us what's possible. And the mice tell us we can fully cure an adult, or we can't at all cure an adult. So that's contradictory. So what we know is that you can treat many of the symptoms at any age and let the humans decide what that's going to be. And I have every optimism that every age will see tremendous benefit because of that. And so how do we prepare for trials? There's so much going on. And I'm going to go through each one of these so you don't need to read this slide on all of the things FAST is doing. And these are really what I'm focusing on is where is your funding going? What are we paying for? What are you paying for? So let's talk about each one of these things as we prepare for clinical trials. So we need to create an animal model and cell lines for preclinical testing and de-risking. So we have funded the creation of the Angelman syndrome rat model, the large deletion rat model, which is a much higher species than the mouse in order to test behavior, to really understand what changes in Angelman syndrome, and that was supported out of UC Davis and USF. And we fully funded this program, and that has been incredibly impactful and really helped people to start doing their drug testing in the rat model, which is a better model than the mouse in many diseases. We've created a pig model of Angelman syndrome, which Dr. Dindo will talk about today, which is unbelievably impressive, and I don't want to give it away, so I'll let him talk about that. But these little pigs look a lot like my little Quincy. I can tell you that. We just created a large deletion mouse model that is not just the UBE3A deletion, but it's a 5 megabase deletion. So it's missing the other 10 genes. So we can figure out what do those genes mean and how are they impacted. And then we meet another mouse model that's missing those 10 genes, but not UBE3A. What phenotype is left? What behaviors are left? What does it look like when a child gets UBE3A back, but still doesn't have those other genes? Because they're half missing, right? The UBE3A is fully missing in neurons, but the other genes are only half missing in neurons. So they may not have as much of an impact because of that once they get the UBE3A back. And we want to see if a mouse can predict that and help us understand what other treatments will our kids need if they need anything else. We've created cell lines out of Yale, human iPSC cell lines, thanks to so many of you who donated your child's blood in order to create these neuronal cell lines that are now being distributed across the world to many different pharmaceutical companies and academic partners. And those are actively at work in many different labs, your kids' cells, to be able to test different drugs on how they work in the cell lines. So thank you to all of you who volunteered those lines. We also are funding this landing pad model, which is an iPSC cell line. So basically, Dr. Yang Wei, who's going to talk about his line, has given his lines to Dr. Albert Kyung, who's going to talk about his landing pad, and ultimately showing how he can really try to mimic all of these different genotypes and test drugs on them to really understand the impact of the extra genes outside of UBE3A, the extra RNA in UPD on both copies of the UPD genotype, and really understand what we need to do about all of those other things outside. and then they're creating organoids or mini brains in order to test the communication between neurons of every genotype, so that we can really look in a deeper way without having to necessarily have animal models or test it in humans to figure out where we're seeing impact. And then Dr. Kyung is also doing reporter cell lines in order to be able to look at the activation of the paternal gene and let it turn bright red or let it turn bright green, right? Bright is good, which means the drug turned it on. So that's a really nice way for us to be able to screen and do high throughput screening for drugs, which we've been doing in cell lines in animals for a long time. We just recently in 2020 funded this collaborative brick and mortar program at UC Davis, which is really an infrastructure program or a core. So we funded an Angelman syndrome core. And ultimately that core is meant to provide a fee for service to support preclinical drug testing for many different pharmaceutical companies and academic partners. When you see so many different studies contradicting what they can and can't rescue in an animal model, a lot of that weakness is the fact that people that are not experts necessarily in testing the animal model are doing a lot of the drug work. And they're trying to see what they can rescue in their hands, and sometimes it's contradicted to people that know the model well, because the model is very, very finicky. And so these people know the model incredibly well. There's a couple of institutions around the world that know the model well, and this is one institution that knows it incredibly well, and they can basically take the drug from anybody, test it in their animal models, the mouse, the rat, the cell lines, whatever it is that they want, and then they can spit out the data to tell the company, this drug works, this drug doesn't seem to work, we're seeing rescue in this behavior but not in this behavior, and we can trust those results. And so they've actually been able to come very far through all of their expertise in neurobehavior, molecular biology, drug screening, biomarker assessments, and really looking at these animal models and cell lines. And so in 1 year of funding, they have had 5 pharmaceutical companies come through and support their drug development through the idea that we had this infrastructure, that they didn't need to do it in their house, they could do it in our house, because that saves them time and money. And so that is how we advance drug development, which is so incredibly impressive. And I'm very proud of that. They're an amazing team. And then we also really had to work through developing InSync, which is the International Research Council. And that was really to become an international collaborative where we can bring global opinion leaders together from neuroscience, genetics, behavior, clinical medicine, industry, business development, regulatory sciences, and biomarkers and outcome measures, and do a gap analysis on Angelman syndrome. What are we missing? What science aren't we funding? What regulatory issues do we have, and how do we advance all of the drugs together for everybody as quickly as possible? And we were able to really grow all of these translational research initiatives through these experts to really dive in and critique what we're doing to ensure we advance all shots on goal. And that is really helping to accelerate drug development, both from preclinical all the way through to clinical, and hopefully through to approvals for everybody. Angelman syndrome has 2 natural history studies that were funded since 2006. This was funded through the NIH and the FDA, thanks to the incredible team of clinicians that we have in the United States, that Wen Han Tan and Anjali and Ann Wheeler and Lynn Bird and so many other of you that are working throughout the United States in order to support this initiative. But this first natural history study, they had over 300 patients, and since 2018 in this natural history study, I think it actually started in 2017, they have over 150 patients enrolled. That is thanks to you for lugging your kids to all of these hospitals to get this data in there, because we need to understand their trajectory without treatment so that we can show what their trajectory looks like with treatment. And that is so incredibly important. And thanks to all of our FAST global community, who they are all now creating their own natural history studies in their language, in their country, they are going to understand what things look like and how they can use these tests that will ultimately be in clinical trials in their language and in their culture. And so this is incredibly important work that we're very excited to support. The Global Angelman Syndrome Registry that was funded out of FAST Australia, that has been incredibly impactful and now has nearly 2,000 individuals enrolled. We encourage all of you to go to the Global Registry so people can understand where are patients, what state are they, what is their demographic, what country do they live in, where are they around the world, so we can know where is the best place to direct people for clinical trials. And so this is incredibly important. We have patient-focused development efforts, and so this is really, really important, and this is what the ABOM is all about, the Biomarker and Outcome Measure Consortium. What is meaningful to you and your child? It doesn't matter what pharma cares about. It matters what you care about, because a drug won't get approved if it's not meaningful to you. But we need to figure that out. So really, Roche was very supportive with the ABOM in developing a disease concept model. And then what we did in 2018 is we did a survey of 332 families. And what we found was the most meaningful impact was if we can make a change in communication and speech. Next was seizures, and then gross and fine motor skills, cognition, and activities of daily living or daily life skills. And so if we can see changes in these things, that would be important and meaningful to our families. And that's what people need to know as they develop their clinical trials. We went to the FDA, we told them this, we talked to them all about it, and that's when they encouraged us to say, you need an endpoint that supports communication in Angelman syndrome, and stop using all of these endpoints that really can't capture your kids, because your kids can't be tested on these measures that require speech. And so that's when they encouraged us to work with the Duke team, Dr. Bryce Reeve and Christy Ziegler, and we developed the ORCA, which is the Observer Reported Communication Ability Measure, which now, knock on wood, nearly every pharmaceutical company is using in their clinical trials. And that is the #1 endpoint for communication so far that we have seen in Angelman syndrome, which is incredibly promising, and hopefully all the regulatory agencies support that. But what I can say is that the FDA supported it enough that they actually gave a $2.5 million grant to the Duke team to help develop this for many other neurodevelopmental disorders, 14 other, thanks to the support of Terry Jo Bichelle, Combined Brain, and Duke University. And this all started with this fast-funded grant for Angelman syndrome. So again, we're here to lift all boats, and if we can help every other disease, we want to do that along the way. No offense, Angelman comes first, but we will lift you all if we can. And so that's the ABOM. And what the ABOM is able to do is prioritize what's important to families and what's important to pharma. And this is a dashboard that we made. Look at all of these endpoints that we can use in Angelman syndrome. Our pharma companies together, 28 of them, came together and prioritized what is important for us based off of what is important to you. And this is when we talk about the ABOM, this is what we're doing. We're working really hard to say this is what's meaningful, this is what's measurable. Then we have to combine, if it's meaningful but not measurable, how do we make it measurable? And FAST just invested $1 million a year into making it measurable so we can show it's meaningful. And we can't get approved drugs if we don't do that. So this is what we do at ABOM, and we're working really, really hard to make this dashboard not only available for Angelman syndrome and advancing all of these for all of our clinical trials, but then leveraging that to many other rare diseases as well, so they don't have to spend the money that we've just spent, and they can grow it for their specific disease as well. So what's our goal? Each therapeutic platform can potentially benefit a different target. Different individuals may benefit from different approaches in different ways, and we need to investigate each option thoroughly. We need to de-risk each option so it's appealing for pharmaceutical companies to take that forward to clinical trials. The more we de-risk it or make it appealing and show that it's closer to a potential success, the higher chance we have of a collaboration for a pharmaceutical company to take that through clinical trials, and the more attractive that is for commercialization and potentially approval. And that's how we get global patient access, is approval. And that's really where we have to be focusing. And so when we think about the roadmap, which I know everyone waits for this slide, it's coming. When we think about the roadmap and we think about where we were and where we are and all the programs that are moving ahead, really this is what it looks like. We start with basic research, we move to drug discovery, we get to preclinical, and then once we have a human candidate, we have to take that through toxicology studies called IND-enabling studies in order for them to approve that that can go into a human. Once that's approved that it can go into a human, it can get submitted to the regulatory agencies, and then it can go into clinical trials. Phase 1 is safety typically, phase 2 is safety with some efficacy, and then phase 3 is a lot of safety and really having to prove that it's efficacious and meaningful and impactful before you can get an approval. And so drug development really starts like this. And for every here's some statistics. For every 15,000 candidates that are screened and evaluated, 5,000 get through IND-enabling studies, 10 get into a clinical trial, and 1 gets approved. So we need to fund every single shot on goal and de-risk it as far as we can to get as many in this space so that 1 becomes a lot more than 1. Because we don't know what's going to work for your kid or my kid. We don't know. What genotype, what age may all be different candidates. We need to support all of them, and that's what FAST is here to do, is to support all of them. And so really the Fast Fire team that started in 2013, which I already talked about, thanks to the support of the Azut family, as well as the leadership of Dr. Ed Weber, Dave Siegel, Scott Dindo, Ann Anderson, Jill Silverman came on later in the course, and Kevin Nash, we were able to really advance so much basic research in drug discovery and find candidates that needed to go through to preclinical. And then the infrastructure grant that I just talked about, which we funded in 2020, has accelerated that preclinical space from candidate to human candidate. And then we love this red box, right? This is like the famous red box. We want to get through IND-enabling studies because that means we're getting to a clinical trial. And our goal has always been get to a clinical trial to get to humans, leap over mouse and get to humans. And that's really what our goal has been. And so this is where we were 6 years ago. When I joined the community, when Quincy was diagnosed 6 years ago, we had an AAV, an ASO, an artificial transcription factor program, and 2 downstream targets. So we were sitting in this drug discovery preclinical space. 3 years ago, this is what the pipeline looked like. So we were moving into clinical trials with some downstream targets, that was Ovid Therapeutics, and we were moving ever closer toward that preclinical space. 2 years ago, this is what it looked like. So we're moving rapidly. And suddenly things are kicking off and things are really accelerating. This is where we were last year. And if you want to see where we are today, this is what it looks like. And this is incredible how far we have come in such a short period of time. And with that certainly comes a lot of work and a lot of expense and unfortunately some failures. And Ovid Therapeutics, their trial failed this year, and that was devastating for so many of you, and we know that. But we learned so much from that trial. We learned so much about what it takes to get through and to clinical trials, and we're so grateful to all of you who participated, as well as Ovid for their efforts in that trial. We learned a lot. But what I want to really focus on is all of these marks. Every one of those black pins is a FAST-funded program. We have accelerated so much because of all of you who have invested so much time, effort, and money into allowing us and trusting us and all of our scientists to do their job and accelerate translational research toward human candidate. And all of this is because of you guys. And I hope you are proud of yourselves, because I certainly am proud of you. So we think about the timelines and what that takes. It costs, as you move forward, it costs more and more money. And so we're really focused in this area right now, and it's incredibly expensive and takes an incredible amount of time. But while we're doing all of that, there's a whole lot more in the preparation that we talked about, right? I'm just talking about drugs. What about everything else for drug approval? So what does that take? There's a lot of work that goes into approving a drug. Creating animal models and cell lines, really talking to registry, getting a registry and doing natural history studies, understanding the disease concept and developing endpoints, engaging with regulatory, helping with clinical trial protocol considerations, diagnosing patients and doing newborn screening to diagnosing them as early as possible. And so where are we? This is where we were in 2015 when Quincy was diagnosed. And this is where we are today. So we are not only advancing drug development, but we're advancing everything it takes in order to get through to patients in the end. And we are moving this ever faster, as fast as we possibly can, because time is of the essence, and we all know that. So this is the space that we were, and this is the space now that we need to go. And we're moving with the times, right? We have to move with the times, and we're so proud that those times are accelerating. So gene therapy has a lot of work ahead of it, but it's moving very quickly. And everything that I just talked about is we really need to ensure with gene therapy that we can get into enough neurons to support it. We're turning on the paternal copy of that gene. We have various different targets with the different companies, different dosing frequencies, and we need to figure out those candidates that are going to be best for each patient. We're looking at downstream targets. What do we consider outside of UBE3A? Clinical trial readiness, we're really supporting the ABOM and understanding what's minimally important clinical change for differences in our children as they see drugs and as they see improvements, what's meaningful. We need to support all the clinical trial sites, both in the United States and globally, to ensure that we can get everyone where they need to go. And we need to continue really to support drug discovery in every way, because every day there's new technology. So we need to think about the then, the now, the later. We have to go forward, we have to look behind. And so this is an incredible process that's really, really difficult. And we really need to think about how we support global patient access. We're doing the economic burden of disease thanks to the collaboration of the Angelman Syndrome Foundation and Lauren Hoffer, one of our board members who has been working so hard on the economic burden of disease with Amanda. So both of you, I'm so grateful. And so really when we think about the cure, we collaborate, we understand, we ready, and we expedite. And I hope I showed you today how we really work hard with your donations to do that. Numerous potential therapeutic candidates are coming forward. AS is unique with many promising results. We focus to identify gaps and accelerate all these strategies to know that we have multiple shots on goal for every genotype, for every age, and for every country. And that's the most important thing to our community. So if we go back to this and we think about where we are and where we need to go, you could see all of the lines are lining up and this is where we are. But ultimately, like I said, this is where we need to go. And when we think about the next step in this journey in drug development, we need to keep up with where we started and this rapid pace that's happening, which we should all be so very proud of. We're really been humbled to support so many years all of the things that FAST has done, but FAST is really ready to take the next leap and to really jump into this space so that we can help all boats, all of these programs we want to help and we want to support. So I want to really let everybody know, and I'm really honored and humbled to share with you that we are bringing on two very, very special, important people into the FAST community and into the Angelman syndrome community. And these two people are pretty incredible. They've both been involved for over 2 decades in drug development. They both focus on rare disease throughout their entire career, and all they have done is essentially accelerate drug development for disorders like Angelman syndrome. And they'll speak to you a little bit more tomorrow, but I just wanted to introduce them to you today. They really have been an incredible asset and will continue to be an incredible asset to the Angelman syndrome community. And they're going to support our academic researchers, all of our industry partners, every organization around the world supporting Angelman syndrome, and all of our community partners in order to reach one single goal, which is to bring meaningful treatments to every child living with Angelman syndrome. So we've crossed this red bar, and ultimately, it's important that we realize as we continue to accelerate and work back here, we bring on people that can accelerate and work up here. And so I'd like to introduce to you Jennifer Panagoulis and Lance Cowell, who should stand up right there. So if I can just tell you a little bit about them, and I'm going to read their bios because I want to do them every service, and I want you to talk to them throughout the rest of the day as well as tonight and tomorrow because they're amazing, and they're here for you. They're here for every partner. They're here for every industry member. They're here for every organization. They're here to support Angelman syndrome to the next level. So Jennifer has extensive experience working with the FDA and other global regulatory agencies in development, review, and approval of multiple drugs for patients with rare disease and has a broad proficiency in the pre-competitive space of developing biomarkers and outcome measures. She has been involved in numerous programs from preclinical stages of development through all aspects of IND-enabling studies, through all phases of clinical trial design, setup and execution, and then through numerous approvals. Many of you know Jennifer, as she has been supporting the Angelman syndrome community for the last 4 years through being an integral participant in the ABOM, as well as supporting Genetics Biotherapeutics from its inception. Jennifer is a dear friend. She's an amazing, incredible colleague, and maybe one of the most impressive individuals that I've ever met when it comes to drug development. She is an absolute unicorn, and we should be so excited that we have her for Angelman syndrome, which is incredibly exciting. Aside from all of this, the most important part of Jennifer's resume, actually, is that she is the aunt to a 19-year-old beautiful girl named Samantha who lives with Angelman syndrome. If you can put my slides back up. I'm not done. Thank you. And I'd like to also introduce Lance. And Lance will talk more for himself tomorrow, but Lance comes to us with experience across 20 different therapeutic areas, most notably being integral in one of the most successful rare disease launches, Spinraza, our holy grail for intrathecally delivered antisense oligonucleotides. For those of you unaware, Spinraza was the first ITASO delivered and approved by Ionis and Biogen. And it completely changed the trajectory for those living with SMA. And most of us live for what happened in the SMA space in hopes that that will happen for Angelman syndrome. After Spinraza, individuals with SMA used to not be able to survive past the age of 2, and now they live a very appropriate length of life and a very essentially near normal life. They no longer die from SMA, which is pretty incredible. It was one of the most miraculous medical marvels in my mind that we've ever seen. And Angelman syndrome, we hope, will be the next one. Lance has spent 14 years at Biogen. He supported or led various drug launches in the US and Europe and globally. And he worked in ranges of different roles at Biogen as the head of multiple global market affairs and functions, as well as the VP of Global Commercial Excellence and Operations. He's a total force to be reckoned with. I've now been working with Lance for a few months, and he is incredible. He knows that knowing that we have him on our team and on our side supporting nothing but Angelman syndrome has given me a tremendous amount of peace. Knowing that all players out there can benefit from his awe-inspiring work ethic and his mission to cure Angelman syndrome, just like the rest of us, is really what makes me so excited for the next steps of FAST and for all of the Angelman syndrome community. So Jennifer and Lance, we welcome you to our team. We welcome you to our community. And then finally, I will end it here. So this is just a year in review, which I already mentioned essentially everything through my presentations, but I just want you to see how much we have done in the last year, how hard we have been working. Our head has been down. We have been working incredibly hard for all of our kids, and a lot has happened, and now, thanks to Jennifer and Lance, I believe it will accelerate and a lot more will happen. We want to thank all of our partners and everybody that has supported all of the possibility for making that pipeline possible. Not only our pharmaceutical partners, but all of our research partners, all of our community partners, but most importantly, we want to thank you. We want to thank all of you for all of the work that you have done to keep yourselves afloat at home while out there fundraising for our children. I'm not fundraising for our children like you are. I'm just here trying to be the project manager, organizing to make sure that all of this can happen. This is a team. This is not about one person. This is about all of us because we're a community, and we're a community together. So I'm so thankful for all of you. So we want to also welcome community leaders. We have so many community leaders that have been supporting FAST, and many of you have approached us on how you want to further support FAST. So here are opportunities. Please come and talk to us throughout the entire weekend where you feel you can be most appropriately fit if you want to be a community leader and play a bigger role at FAST. Whether that's through really helping bring an army together to accelerate our roadmap, whether that's being an ambassador and helping to spread the science around the world through social media and through various different methods, or whether that's just really supporting fundraising needs and helping other friends fundraise who want to but don't know how. So we want to utilize all of you to help our community because we are one community and we can use your support. We want to bring you in on how you can help us as well. So this is a team, and we're so excited to welcome you to our family. So with that, there are many disorders that will never be cured in our lifetime, but I guarantee you Angelman syndrome will not be one of them. Remember, knowledge is power. You need to understand in order for you to make decisions. And we're here to help you understand, but your doctor are the people that will help you understand the most. So we can guide you with basic understanding, but for decisions for your kid, you need to really be informed. And that information is not only going to come from lectures like this, but it's going to come from your doctors. So surround yourself by that and really realize that we need you all to join the mission to cure AS because it's coming and we've never been closer, and this is a really exciting time for our community. So let's work together and make the impossible absolutely possible. Thank you.